Welcome back. Now we have something very special in store for you now. We all like to hear positive stories, especially at the moment. And our next two speakers today definitely have some positivity to share from their clinical pipelines. Our first speaker is Xiaokui Zhang, who is Chief Scientific Officer from Celularity, who are currently developing placenta-derived off-the-shelf allogenic cellular therapies. She is the company's Chief Scientific Officer and will be giving us an update on the latest developments in the pipeline. But very quickly, before we jump into our first presentation, one quick announcement. Thank you, everyone, for adding the roundtables to your schedule, and we're pleased to see so many of you are interested in joining those. A quick note, though, please, because those sessions are space limited, if you're no longer able to join one, please could you remove it from your calendar so your slot can be opened up for somebody else. Thank you. Right then, over to you, I think now, Xiaokui.
Great. Thank you so much, David. I'm Xiaokui Zhang, Chief Scientific Officer of Celularity. I'm so grateful to the organizers to have this opportunity to speak at ATC about the allogenic placental-derived NK cell therapy. Next slide, please. So here is a forward-looking statement. And next slide, actually, I'd like to talk a little bit about who we are, what we do, before we get into the clinical program. So Celularity is a clinical stage biotechnology company. We're developing what we believe is the next evolution in cellular medicine, which are the off-the-shelf allogenic therapies that derive from the postpartum human placenta. We, over the years, have established IMPACT technology, stands for immunomodulatory placenta-derived allogenic cell therapy, so which capitalizes on the benefit of the placenta-derived progenitor cells. So those progenitor cells are so abundant and evergreen as a starting cell sources for a variety of the shelf products. So the stemness and expandability, which we'll talk about a little bit that at the later slides, that really is associated with the fundamental biology of the source material derived from the placenta. And we believe this is very important for the cell therapy to be able to persist and proliferate after the patients are infused. And you'll see some of the data that coming up. And also some of the immunogenic properties, such as the cells being naive, allowed for improved safety profile for those therapeutic products. Again, you'll see some of the data that from our translational studies. So our preclinical and the early clinical data demonstrate a variety of the biological activities that are suitable for those therapeutics across the various areas, including hematological malignancy, solid tumors, infectious disease, and degenerative diseases. Next slide, please. So actually, Celularity that really derived from a company called Ensogenesis, founded by Dr. Robert Harari about 20 years ago. And that company merged into Celgene at the end of 2002 to build its cellular therapeutics division. So we actually really benefited from Celgene's 20 years of the investment in research, development, and also getting the capacity together that required for the manufacture cellular products at scale with high consistency and reliable quality. So just recently that we moved into this purpose-built, really nice facility that located in the northern New Jersey, ready for the commercial scale manufacturing. And also it includes a research, QA/QC, clinical operation, biorepository, all under one roof. So we have about 150 scientists and engineers and technicians that are really working hand in hand, developing those innovative allogenic cell products. So next slide, please. So here, let's focus on the first allogenic placental CD34 derived NK cells. So those are the cells that are referred as a CYNK001, and those are the cells that manufacture utilizing a proprietary process that started with CD34 cells that isolated utilizing the Clinomax and undergoing the proliferation and differentiation, and then finally that into the lymphocytic lineages. So those cells are highly scalable and currently investigated in several phase 1 studies, including AML, multiple myeloma, GBM, and COVID-19. And for today's presentation, we're actually going to focus on the AML clinical trial. So the zinc cells that when we looked into their phenotypic and molecular signatures, that we have found that the zinc cells express a high level natural killer, natural cytotoxicity receptors, and have a lower expression of the CD16 and KIR, that indicating that they have the immature characteristics when compared to the peripheral blood-derived NK cells. And we believe that this relatively immature phenotype that would provide them with the potential to persist and proliferate after they're infused. And also when we compare the telomere length of the CYNK001 cells with peripheral blood NK cells, so we have seen a much longer telomere length, which we believe is associated with the starting material that we utilized for generation of this product. Again, this would support some of the longer persistence and proliferation data, which you'll see in a minute. So the zinc cells that, as I mentioned, that express a very high level of the natural cytotoxicity receptors. So we actually have seen very high in vitro cytolytic activities against a broad range of the tumor cell lines. And aside from the high tumor killing, we also have seen the high production of the interferon gamma production. But aside from the high level of the activation that we have seen, there's a low to no expression of the exhaustion markers such as the PD-1, TIGIT, LAG3, or TIM3 that being detectable both in vitro and in vivo. Next slide, please. So this slide summarizes the key preclinical data that demonstrating the high anti-leukemic activity demonstrated by CYNK001 cells. So on the left side, you can see the very high killing that demonstrated utilizing CML, AML, or multiple myeloma cell lines at a different effector to the target ratios. And in the middle, that's showing in presence of those tumor cells, a very high level of the interferon gamma production that being observed. And this is one of the cytokines very important to prime and activate adaptive immunity, such as APCs and T cells. So this is how the NK cells potentially work hand in hand with adaptive immunity. And on the right side, as you can see, that the high level of the killing against the primary blast cell was demonstrated utilizing the AML, the primary samples. So this is basically the summary that's supporting the investigation of the CYNK001 cells in several hematological the indications which we'll talk about. So next slide, please. So our first in-human clinical investigation of the CYNK001 was in the relapsed refractory AML patient population. So this was a dose escalation study. The patient that actually received anywhere from 1 to 6 prior lines of the treatment were first undergoing the preconditioning with the cyclophosphamide and fludarabine, and followed by a single dose of a CYNK001 cells, and together with up to 6 doses of IL-2. So in this study that we have seen the zinc cells that are very well tolerated in this heavily pretreated AML population. There's no dose limiting toxicity, no GVHD, and no detectable HLA allo antibodies. That's suggesting those cells really have a very low immunogenicity associated with it. So we have enrolled 11 AML patients and 10 of them treated with zinc cells. And out of the 10 patients, 8 of the patients were efficacy evaluable. So we actually have seen a very encouraging clinical activity in those 8 patients. The one patient actually entered partial complete remission at the day 21, and the other patient that achieved the morphological leukemic-free status at the day 14. So let's take a closer look at some of the translational correlative studies that's shown at the next slide. So just to remind the audience, the zinc-001 cells were infused in the AML patients in the mismatched manner. Therefore, we're able to use either the zinc-001 HLA antibody or patient-specific HLA antibody to tell the NK cells apart. So the upper left corner showing how we actually were able to utilize the flow cytometry to detect the infused zinc cells, and those are the dots in the box. So as you can see, the zinc cells that were detectable up to 28 days. So utilizing the multi-parameter flow panels, we actually were able to see those zinc cells not only persisted, they also show the sign of a further maturation, the evident by increased by CD16, pancreas, CD57, and also the fact they were actually positive for Ki67, showing all the way to the upper right side, that suggesting the zinc cells not only persisted, they matured, they also proliferated. So through the entire persistence period of the time, we didn't detect any exhaustion. So that was evident by the lower left corner, lack of the expression of the PD-1, TIM3, or the LAX3 expression. When ex vivoli activated those persisted zinc cells that were able to retrieve from the patient sample, and we saw those zinc cells retained a very high level of the effector functions. They still have the ability to produce the granzyme B, perforin, and interferon gamma. And as I mentioned earlier, that through the detection of the persistence of the cells, we didn't detect the allo HLA antibody at all. So this basically is the scientific data that's supporting some of the clinical activity that we have seen, and also supporting the younger and the cells that potentially with the stemness had a better persistence of proliferation, and which are required for those cells to achieve the therapeutic benefit in those patient population. So next slide, please. So I'd like to wrap up my presentation by showing how that Celularity right now is positioned moving forward. So zinc cells, in addition to the AML cells, are also evaluated in several other clinical indications, including glioblastoma multiforme. And as I mentioned, that we're actually treating COVID-19 patients at the current time. In addition to the unmodified NK cells, we also have the genetically modified NK cells as referred Zinc-101, that for a potential combination with Herceptin in HER2 positive gastric cancer. And we're ready to file the IND this year. So in addition to that, we also have allogenic CAR-T, the product coming up targeting CD19 positive B cell malignancy, and another MSC-like product referred as Apple that evaluated in several degenerative diseases. So very excited to have this opportunity to present here, and we're actually really looking forward to some of the Q&A interactions at a later time. So with that, thanks for your attention, and David, go back to you.
Super. Thank you very much indeed, Xiaohui. Incredible progress. Thank you for sharing it. And I too am sure the audience will have plenty of questions that they want to put to you. And there will be an opportunity to do so right after this session in the Zoom Q&A session. Our next speaker is Kimberly Noonan, Chief Scientific Officer at WindMIL Therapeutics, who are currently developing bone marrow-derived memory T-cell therapies for cancer patients. So, Kimberly, unwrap that for us. Over to you.
Thanks so much, David, and thanks to all the organizers for inviting us here today. We're a little remiss not being in Miami today, but it's nice to be here with you on our virtual world now. Next slide, please. So I want to talk to you a little bit today about WindMIL Therapeutics. So we are developing novel insights of bone marrow biology into treatments for cancer patients. And we see this as being one of the next generation of cell therapies for cancer patients. We've been working on MILs for quite some time now at Johns Hopkins University before we spun it into WindMIL Therapeutics. And we have an exclusive position in MILs at WindMIL Therapeutics. Process we brought over is proprietary and scalable. Transform and expand experienced bone marrow T cells. And we're going to talk a little bit more about that biology in a minute into products. The natural qualities, the biological qualities of MILs have advantages over other cell therapies such as TILs. And our platform supports both unmodified MILs as well as gene-modified or CAR-modified MILs. We have a very strong IP position for composition, process, and use patents, and a lot of deep know-how behind the work that we're doing. We have a solid tumor focus right now with key readouts in the next 12 months. We have a clinical trial open in non-small cell lung cancer. It's second and third line refractory relapsed non-small cell lung cancer, and it's in combination with Nivo with a collaboration we have with Bristol Myers Squibb. We also have a great CAR-MIL program in the preclinical setting that we're going to talk about a little bit at the end as well. In both heme and solid tumors, we're looking at this program. Next slide, please. So if we think about where T cells come from for cell therapies, in general, you see them coming from two sources, the tumor, so TILs, which are antigen specific and antigen experienced, but have more of an exhausted phenotype in trying to impart their antigen specificity in the tumor microenvironment. The peripheral blood in general lacks broad antigenic specificity, and that's imparted into the cells using CARs. And so that's where you get most of the cells that are used for CAR therapy. Instead, we're focusing on bone marrow T cells. It's a cell source that's unique to WindMIL. These cells are patient-specific. They're broadly antigenic specific. They're highly cytotoxic and highly persistent after we activate and expand the MILs product. So what's important about the bone marrow? Immunologically, the bone marrow is a reservoir for antigen-experienced memory cells. And these cells we've shown in multiple myeloma and other hematologic malignancies, but we and others have also been able to show the existence of these cells in solid tumors. We have data in non-small cell lung cancer, breast cancer, head and neck cancer, GBM, but other groups have published as well in cancers such as breast cancer and GBM. Some of the most important work coming out of Führer's and Schumacher's group in Germany, showing that the most tumor-specific T cells in breast cancer patients are actually in their bone marrow. We can go to the next slide, please. So this is a schematic of our manufacturing process. It's a proprietary manufacturing process. It is bone marrow is taken from the iliac crest of the patient. It's a very friendly process for the patient, 30 minutes in conscious sedation to take the bone marrow out. This bone marrow contains T cells that are antigen specific. This bone marrow is transported to our contract manufacturing facility, which is Cognate Bioservices in Memphis, Tennessee. It undergoes a 7 to 10 day proprietary manufacturing process where we activate and expand the cells and transform them, and then undergo typical testing for cell therapy products. They're then transferred back to the manufacturing facility, and in around 23 days from harvest until infusion into the patients, we're able to treat our patients. Again, a very patient-friendly procedure, a 30-minute bone marrow collection under conscious sedation. A reliable procedure. We have over 90% success manufacturing, and a very scalable, cost-efficient procedure as well. Next slide, please. So we have been working, as I said, on MILs for several years now at Johns Hopkins University. So we have a bit of safety data. And so if we work from the bottom up, that's sort of how the progression of our studies have gone. We have had data in a phase 1 and a phase 2 study in high-risk multiple myeloma, and have seen a very safe, favorable safety profile on no high-grade CRS in any of our patients, and some interesting early clinical data as well. We have an ongoing study at Hopkins now in patients in an allogeneic transplant setting. This is donor-derived MILs, if you will. We've had no cases of GVHD observed, very important, and no CRS. And we've seen some early evidence of clinical responses, 3 CRs and 12 patients achieving stable disease or better in a 14-patient subset. And non-small cell lung cancer, that's the current study that's undergoing at WindMIL. It is initiated, it's MILs only monotherapy for 2 patients for a safety cohort. And then we moved on to MILs plus nivolumab for a total of 20 patients treated with MILs plus nivolumab. Again, in the safety profile, we have no CRS, and a very favorable safety profile allowed us to move into the MILs plus nivo arm. And again, a very favorable safety profile thus far. We have 4 patients at stable disease, 2 patients out a year, which is very exciting, and some encouraging survival data overall. Next slide, please. So this is the study that we're running right now. Again, it's relapsed refractory patients with non-small cell lung cancer. It's locally advanced unresectable, and patients who have previously progressed on PD-1 containing regimens. Flu-Cy as a PrEP regimen, MILs plus nivo, and then our primary endpoint is overall response rate, and then follow up for overall survival as well. We are going to continue to collect data over these next 12 months. We're very excited for these 12 months because not only will we have expanded clinical data, but also our correlative translational data as well. Next slide, please. In the background, we are positioning ourselves to move into other solid tumors as well. And we presented some work at SITC last year showing that we've been able to activate and expand cells, MILs, in many solid tumors. We have a process for this now for breast cancer, glioblastoma, head and neck cancer, and prostate cancer. And we've been able to show that the expanded MILs for patients with all of these solid tumors have antigen-specific, shared antigen-specific T cells. It's consistent data from our myeloma and non-small cell lung cancer cohorts. And we've also been able to continually show that matched peripheral blood T cells do not show this specificity in our work. And so what we see on the right-hand side of the slide is the ability of MILs, and we're looking at CD4 and CD8 subsets, to produce interferon gamma to shared antigens associated with the tumor type. And you see the background, sorry about the background with my cat. This is the real life that we're living right now. But you see the matched peripheral blood cells from those same patients not producing gamma, while you see nice gamma production in all the solid cell types with the MILs. Next slide, please. So the other thing that we're working very hard on at WindMIL is our gene-modified platform, what we call CAR-MILs. And we didn't bring a lot of data here today, but we are going to talk a little bit about what we're about to hopefully publish. It's being submitted, as well as a relationship that we have with Penn to continue to study this data. But we have some biological background of MILs. They have a very favorable metabolic profile. We've been able to show long-term clinical persistence and enhanced expression of chemokines and chemokine receptors in these cells. And the advantages that you see is that these cells are consistent with memory effector phenotypes because of the metabolic profile. They maintain clinical effectiveness with their long-term persistence, and we have published that and presented that in the past. And with the increase of chemokines and chemokine receptors, they can traffic. Particularly, MILs overexpress the chemokine receptor CXCR4. It allows them to traffic to high levels of stromal-derived factor 1 found in the bone marrow, but also importantly found in solid tumor stroma. So if we take these biological advantages of MILs and then think about how that would play forward into a CAR platform, it does very nicely show advantages of CAR-MIL compared to CAR-T. So we see superior cytotoxicity with CAR-MIL, and it's a persistent cytotoxicity and very robust at extremely low effector to target ratios. We look down to 1 to 100. The native TCR of our cells remains operable, and this we think will be important to cover antigen escape variants with CAR therapies. We see a superior cytokine profile, very high PSI score, and this high PSI score has been associated with clinical responsiveness in these patients. We've also seen in vivo models kinetic advantages of CAR-MIL compared to CAR-T. So CAR-MIL are killing faster than CAR-T cells, and this could lead to decreased dose on overall persistence of these cells if given clinically. Next slide, please. So we have entered into a collaboration just before COVID hit us last year with the University of Pennsylvania under the direction of Carl June to really move this platform forward, utilizing Penn's excellence in CAR-based therapies along with our deep understanding of MILs. And the collaboration directly is comparing CAR-T generated by Penn with CAR-MIL generated at WindMIL, leverages Penn and WindMIL's expertise and resources as well as our science, and we retain full control of on all rights to our CAR-MIL product. This research will substantially expand and further validate our CAR-MIL platform and will allow us to move quickly into a clinical trial in the upcoming year. Next slide, please. So finally, I want to wrap up with our portfolio and our pipeline. So I hope today we've shown you that MILs have broad antigenic specificity. Although we haven't shown the data today, they are highly cytotoxic and they have a very favorable safety profile clinically. And we believe that these cells could be used in earlier lines of treatment. Our CAR-MIL platform enhances CAR-based therapies with all of the attributes that we just shared, and we believe that we can use CAR-MILs in patients who have more advanced tumors. And so our non-small cell lung cancer is in phase 2, and breast cancer probably coming up later this year in phase 1 study. In our heme malignancy session, we will be continuing to look at the data in the allotransplant and moving forward our preclinical work in our gene-modified cells. So I think I just about used my time properly. David, back to you in the studio.
Amazing. Thank you very much indeed, Kimberly, for sharing your incredible work with MILs. Thank you to your cat as well for almost stealing the show, but your work certainly took the spotlight there. And also thank you to Xiaokui for sharing her very exciting pipeline developments. If you have any questions for either Xiaokui or Kimberly or her cat, then do hop on to the Meet the Speaker session where you can interact with them face to face. After this, on this track, we will host our Tea and Tech series of technology demonstrations. So see you there, and don't forget your tea.